Okay, today we want to talk about inheritance. And inheritance involves genetics. Heredity or hereditary traits are passed from the genetic material of the female and the male involved with reproduction, and this is known as genetics. The genotype of an individual is the actual chromosome makeup of an individual, and that leads or is expressed as a phenotype. So we have genotype. That's supposed to look like a G there. Versus phenotype. So the genotype is made up from the genes, and the phenotype is the expression of the genes. Now, humans have 23 pairs of chromosomes, and one of each pair is provided by the mom, and the other pair provided by the dad. On a given chromosome, there are many genes. There can be as many as several hundred to several thousand genes on a chromosome pair. So the gene is a sequence of nucleotides, and they're arranged in a helical sort of fashion known as DNA. And if the DNA message is going to be delivered within the cell, it creates a messenger RNA, and that's going to code for a particular function, usually the production of a protein, like an enzyme, for example, or maybe a hormone, that sort of thing. And those genes create different DNA sequences, different proteins, and that's what creates a genotype. Humans have somewhere between 20 and 25,000 genes. Usually it's reasonably well accepted that it's in the neighborhood of about 22,000, 22,500 human genes. And we have 24 distinct human chromosomes. We have 22 pairs. That are described as being autosomal. In other words, they look similar. So under a microscope within a nucleus, they may look something like that. That's a pair of chromosomes. There may be another pair that's a little bit different shaped. But again, each half of the pair looks the same. So that's where the autosomal term comes from. Then we have an X chromosome. And it's a pretty large chromosome. It's got lots of genes on it. And the Y chromosome looks different. It's quite a bit smaller, and it has less genes on it. But crucially, it has a particular gene, which we're going to talk about a little bit later, that's going to be the sex-determining gene. So in a chromosome, there can be as many as 50 million to up to about 250 million of these nucleotide pairs. So you might be generally aware of what these nucleotides are. They're adenosine and thymine, guanine and cytosine. So these are proteins, essentially, that are connected to one another. They're paired up. And in a given chromosome, there's going to be multiple millions of these sequences. And the arrangement is almost always going to be the same in terms of the pairing. The adenosine matches with the thymine. The cytosine is going to match with the guanine. So you get an AT pair and a CG pair. And these repeat themselves in varying patterns throughout the length of the chromosome. So this DNA strand can have millions of these pairs associated with them. And within this long strand of millions of paired nucleotides, there are some sequences that give us our genotype. So amongst those pairings, we're going to find those 20 to 25,000 human genes. So there's a long chain of DNA, and those DNA pairs that are formed are not all contributing to our genotype. They, in certain segments, we're going to get the genes that create that different DNA sequence that I mentioned a few minutes ago to give us our genotype. So the genotype is under the influence of our environment and developmental factors that are going to end up expressing our phenotype. So we may have a long strand with the pairings all the way through it, and a segment of that long strand represents a gene location. And we'll on a chromosome, within a chromosome. That's sometimes referred to as a locus, or if it's plural, loci. And the expression of these genes is dependent on factors like whether or not the contribution from both mom and dad are the same. That would be called a homozygous gene. If they're different, heterozygous. If they're dominant versus recessive. If they're incomplete in their dominance, or if the gene is expressed through multiple allele inheritance. So typically we think of dominant and recessive. So if mom has recessive and dad has dominant, you would have a pairing that would be described as being heterozygous. When we have multiple allele inheritance, there's more than just the two alleles that can contribute to the genotype, which typically is going to change the phenotype. So we'll talk about some of those examples in just a minute or two. Alternate forms of a gene that code for the same trait are known as an allele. So there's going to be an allele contributed from the female and an allele contributed from the male in the reproductive process. And that's going to create a trait and ultimately an expression of a particular trait. Now, when we think about all of this genetic information, what we want to recognize, probably before we get too far along in this kind of a topic, is about 99.9% of our genetic information is common in all humans. So that leaves us with a 0.1%, a tenth of 1%, to account for what gives us a different appearance. For example, the color of our hair, the color of our skin, our height, our disease risk, eye color, all those sorts of things are accounted for in that 0.1% difference in our genetic material. So this can be deceptive, but if you remember that I said we have millions of pairs of nucleotides that make up our DNA strand, and only small segments of that strand represent genetic expression. So all the rest of it, about 98% of that material there, is designed to influence gene expression or take up space within that chromosome. So the rest of the DNA is left to either turn off or off genes, or can be transcribed onto an RNA molecule, or is just a filler in some respects. At least we don't know what else it might do at this stage of the game. Back in the day, when it was recognized that not all of the DNA molecule contributes to genetic expression, some of that in-between DNA was described as being junk DNA. We know that it's not really junk DNA, but it is DNA that doesn't have the same sort of influence over genetic expression that the loci where the alleles from a mom and a dad form a trait. So to put a little bit of that into perspective, most of us are generally aware that chimpanzees, are pretty closely related to human beings. And we share about 96% of our genetic material with a chimpanzee. We share about 60% of our DNA with bananas, if you can believe that. So there's a lot of genetic material that's shared not just between other animals, but also animals and other living organisms like plants and fruits. Okay, so let's take a look. Let's move on to another slide here. Let's look at some of these alleles. So when we say that there's alternate forms, of a gene that are going to code for the same trait, that's known as an allele. So mom is going to provide an allele and dad is going to provide an allele for a particular trait. Now, if the allele is dominant, what that means is it's going to mask any other allele that might be contributing to that trait. So it's sometimes described as the superior or the supreme allele, and it'll mask another allele. Mask or block or cover up, however you want to refer to it as. The other allele type that we want to consider is a recessive allele. And the recessive allele is going to just be the opposite of what we just described. So it gets covered or masked by a dominant allele. So these are the classic sort of basic alleles that a monk way back, hundreds of years ago, recognized, influenced plants. He was a guy that studied plants in a garden, particularly pea plants, and they would have colored flowers. And he noticed that some flowers were different colors. And through experimenting, he recognized that if there was a dominant allele, let's just say it was the color white in the flower, over another color, say green, for example, then he recognized that if you combine a white plant with a green plant, sometimes the green plant presentation was covered over. It was masked by the white. So he concluded that that meant that the allele from the white plant was dominant over the allele from the green plant. And it gets a little more complicated than that, but that was how he got started in recognizing that there was this genetic influence from the parents over the offspring in a reproductive mechanism. So a few other considerations is we can have an expression that's described as being homozygous, and that's when both alleles are exactly the same. Lots of times alleles will be designated a letter depending on the topic that you happen to be talking about. So if we said that the dominant allele could be represented by a capital letter, then our recessive allele would be by the lowercase letter. So in a homozygous presentation, we could have homozygous dominant, and that would look like this. The allele from each parent would be dominant. The same, but represented as two alleles expressing that particular trait. But we could also have homozygous recessive, and that would be designated with the two lowercase letters, the contribution from mom, contribution from dad, homozygous recessive. Then we talk about genes that are heterozygous. And I'm sure we can all figure out what's going to happen with heterozygous. Maybe the dad offers the dominant allele and mom offers the recessive allele. So now we have heterozygous, and the presentation in this example would be the dominant allele. So if we were let's just take something nice and simple. It's not completely accurate, but nice and simple for a visual. If we said that the color brown was dominant over blue, which is true, then we would say that the person that has the double capital A is going to show up as a brown-eyed individual. the person with the lowercase AA would show up as a blue-eyed individual. And the heterozygous dominant, capital A, lowercase a, would show up as a brown-eyed individual, but could be described as carrying, in other words, a carrier of the recessive component to the gene, so that if they in turn underwent a reproductive process at the appropriate times in their life with someone else who had the lowercase a, it's possible they could have offspring with the blue-eyed color because they're carrying the recessive allele. Now, you can also have something that's described as being co-dominance. And in co-dominance, both alleles can be expressed. So that's kind of rare, but it's not completely unusual. And next week we're going to talk about blood and talk about some of the characteristics of blood, and this will be an example of co-dominance. So in that example, we have blood that's described as being type A. We have blood that's been described as type B. And you can have a situation where somebody has type A blood either because of homozygous dominance or heterozygous dominance. And in heterozygous dominance, what happens is the individual does not have a matching allele. So in blood typing, that's a type O person. It's really a 0 for not having an allele to contribute. So we designate that in genetics as a little I with a dot over top of it. So both of these people would test as being type A blood. And we see the same thing in type B individuals. They can be homozygous or they can be heterozygous. But where the codominance comes into play is if a type A person reproduces with a type B person, it's possible that we create a new type, AB. And in AB, you're neither A or B, you're a new expression, type AB. And it's also possible for someone that has type O blood to express as type O. So in this case, we have 3 alleles because we have the absence of an allele, including A and B. So that's going to create 4 different expressions, codominance. Now, in addition to codominance as being an unusual way to express or a different way to express, we can also have something called incomplete dominance. And in incomplete dominance, you may have a situation. The classic example of incomplete dominance is sickle cell disease. That's an anemic disease of the blood where the blood under certain circumstances, the blood cells change shape and they don't exchange oxygen effectively. And so the individual that is expressing the sickle cell trait is going to have trouble delivering oxygen to tissues and having enough oxygen being carried on these abnormal cells for proper oxygenation and removal of carbon dioxide. But in incomplete dominance, neither allele is completely dominant or completely recessive. So we get an intermediate type, we get a different type. So in sickle cell, what happens, and we'll set it up this way. So if we have a healthy individual, that person, so sickle cell disease is described as being a recessive disease. So you can have on your hemoglobin, so here's our hemoglobin, and if we have a normal hemoglobin, we're just going to call that hemoglobin type A. And if the allele that's presented from both parents is hemoglobin normal, okay, then that's somebody that has normal blood hemoglobin. Okay, so we're just designating it with a capital A in this example here. So if it's a situation where we know that the sickle cell trait is recessive, so we're going to designate somebody that has sickle cell with a little s, and both alleles have to be expressed if the sickle cell disease is going to be recessed, is going to be expressed. So this is abnormal. In other words, this is going to be expressed as a sickle cell disease. So these patients are at risk of the blood cell changing shape and getting brittle and fractured and bleeding into joints and causing a lot of pain and discomfort, never mind failure to deliver oxygen to the tissues and that sort of thing. Now, in an incomplete dominance, when you have a mix, so with incomplete dominance, what we're saying is our heterozygous individuals, so they have one contribution for normal hemoglobin and one contribution from sickle cell, okay, then basically what happens with these individuals is they're described as having the sickle cell trait. So just like we mentioned before, they would be described as a carrier. But what we know about these individuals is that under normal conditions, normal environmental conditions, especially within the atmosphere and the air we breathe, then it's normal function most of the time. But under oxygen stress, like going into altitude, for example, then what happens is the signs and symptoms of sickle cell are going to present. And there's going to be an inability to deliver oxygen to tissues and fracturing of those blood cells and so on. So this is an example where as long as there's no oxygen stress on the individual, the blood cells perform the way they're expected to. But under stress, for example, altitude, the expression of sickle cell occurs and it becomes a dangerous scenario for that individual. So there's been some classic examples of this that are more known publicly. Some professional football players, for example, are unable to travel with their team to Denver because Denver is in high altitude. And if they did travel to Denver, particularly if they performed on a football field, so they have that added stress of physical activity with an increased demand for oxygen, on top of the altitude issues in the location of Denver, Colorado. The same would apply to a soccer player, for example, if they went to Mexico City. These are places where the altitude is significant, and they would begin to express their sickle cell signs and symptoms. So most people that have reached that sort of level of physical activity have been tested time and time again for different things and are aware of this. If they're not aware, though, they could have one of these events that occur under that oxygen stress and need to have immediate treatment, which would involve oxygen or a hyperbaric chamber, that sort of thing, to reverse the effects of that sickle cell scenario. So there's another, we mentioned codominance, but sort of part and parcel with codominance is something referred to as multiple allele inheritance. So we can use the same example. We can have type A blood, type B blood, and no type blood, blood O or blood zero. So if we combine any two of these alleles, we could have a parent that's this, a parent that's this, and we could have an AO, right? Which we use a little I for that before. So in this case, and in this case, they present as type A. And then we had our codominant factor here. But the idea here is there's multiple possibilities. So when we look at many, many ways that we as humans present, there's lots of examples of where the gene exists in more than three types, more than two, so up to three and more forms or alleles are possible. So in reality, eye color falls into this category, blood typing falls into this category, and any individual can have only two alleles. So we just have two possibilities in each of these examples, but we have more than one way to present. So if you have two parents, you're going to have two alleles. But in any given parent, there could be more choices available to create that gene or that trait at that location on the chromosome. So our example that we've used here a few times now is our blood typing, but eye color falls into that. Many of our cell markers that identify cells as self fall into this category. We have a group of genes that are abbreviated as HLA genes, and they have more than 100 different types of alleles. So you can have a combination that's going to be very different depending on what allele the dad provides and what allele the mom provides. And that's going to present itself on a chromosome, on a locus of a chromosome to give us a trait, to give us a way that that cell expresses that HLA gene. And if there's 100 different alleles with combinations of between all those 100 different alleles, and then we can have scenarios where there may be more than one locus on a chromosome for those alleles. So now we have even more multiple factors that come into play. And this is a code for a protein antigen in the immune response. So with all these different combinations, it explains how difficult, for example, tissue matching can be. If you're not an identical twin, there's a good chance that these hundreds of different types of HLA alleles are going to create a different gene expression that's presented on that cell membrane, if you can follow all of that. So the point is that tissue matching is going to be very, very different, even between two siblings of the same set of parents. All these different variations that can occur. Eye color is determined by more than one gene, so more than one locus, and there may be as many as 15 that can present under those conditions. So when you have more than one gene, that's described as polygenic. When you have more than one allele, it's multiple allele inheritance. Sometimes you're going to have both. So that can explain why some people's eyes are light blue versus bright blue versus a gray blue, a beigey color, a greeny color, a brown that's light or dark. Multiple factors are going to come into play. Okay, now when we look at our chromosomes, I mentioned earlier that we know that in humans we have 23 pairs of chromosomes. When we were talking about fertilization, we simplified the chromosome number by talking about cells in reproduction that were either full complement chromosomes, we call that a 2N, or half chromosome complement, a single N. So in the autosomal versus sex chromosomes, let's just do this. we have 22 pairs of autosomes or autosomal chromosomes, and that just means that both pairs look the same unless there's a pathology, something unusual, but they typically line up and will be paired up so that they look very similar. There's a locus that connects them. And that can be varying sizes, but the idea is the pairs are very close to being identical. So those are the 22 pairs of autosomal chromosomes, and we have one pair of sex chromosomes. And the one pair of sex chromosomes, they look different. So if a female is two X's, those chromosomes are going to look similar. So we would have, again, a pairing that look pretty similar. But if we have a male with XY allele genotype, then what we're going to notice is that the X chromosome is relatively big and the Y chromosome is noticeably smaller, different. So what happens in spermatogenesis that we talked about earlier, because this is the process of gamete production in a male, we start with our XY cell. And it's going to undergo mitosis and give us new XY cells. But when the one cell, our primary spermatocyte, was introduced to meiosis 1, it gave us two cells that are going to be genetically different. One is an X and one is a Y. And now we have half the genetic material. And then in meiosis 2, those cells go through the second part of meiosis, and we now have two cells that have half the chromosome material X and half the chromosome material Y. So these are the cells that are going to determine the sex of the offspring. The contribution from the male is what determines the sex of the offspring. Females during oogenesis go through our mitosis prenatally, like we talked about before, and then enter into meiosis 1 and stall. But our primary oocyte is ultimately going to go through meiosis 2. And in each case, we're giving off a polar body rather than another cell. So at the end of meiosis 2, we have a single egg, and at the end of in oogenesis and in spermatogenesis, we have 4 cells with half the chromosome material. So in each case, because we started in oogenesis with XX, we're going to always offer an XX from the female side of the reproductive systems. So the primary gene that determines male development is called the SRY gene. And the SRY gene is one of the few genes found on the Y chromosome that is described as being a dominant gene. It must be present in the fertilized ovum for that individual to become a male. Okay, so testes, we get the differentiation into, and so what happens when the Y chromosome is present, it signals the production of some testosterone that promotes the differentiation of the primitive reproductive organs to become testes. If there's no SRY present, or for some reason the SRY gene is not expressing because of some pathology, for example, then by default, the ovaries and the female reproductive system is what develops. So that's how we determine the sex of the offspring based on the absence or presence specifically of the SRY gene found on the Y chromosome, which is going to be produced or introduced or offered by the male reproductive system. Now, these sex chromosomes are also responsible for non-sexual traits. And most of these are going to be found on the X chromosome and absent on the Y chromosome. So in other words, the X chromosome by default becomes the dominant chromosome for these non-sex traits that we're describing. So some of us may have heard of red-green color blindness, for example, situation where most of the time that's going to be expressed in males, but it is also expressed in females. So in this situation, what you could have, because the red-green gene is by default a dominant gene only found on the X chromosome. So if our offspring is expressed with a capital C, so what we're saying now in a female, the female offspring, that's going to be a female with normal color interpretation, right? So not colorblind. So we're talking, in this example, we're talking about red-green color blindness. So what we're saying is that there would be an absence in this individual of the red-green cones. Cones were the receptors for color found in the retina of the eye. So what happens in someone who's got red-green color blindness is that the reds and the greens, so traffic lights, for example, they look basically the same. I've known some individuals that have color blindness, and they'll say, well, you can sort of learn to differentiate between those colors, but they basically look like variations on gray or variations on beige. So it's pretty difficult to determine differences, especially when the colors are in a multicolored presentation of some sort. So what we can have also then is a female who would have the large C, normal, and the small c, recessive. So this is, again, a female who would be described as a carrier of the trait for abnormal color, but otherwise have normal color vision on the basis of the dominance versus the recessive arrangement here. So, but she can contribute to future offspring because she carries the recessive gene. So to have a female who has this color blindness, you'd have to have a colorblind male mate with a colorblind female to give us a female who expresses red-green colorblind. Okay, so that's going to be rare because females will probably, if they understand that their partner is colorblind, they may opt to have a blood test to determine whether or not they're a carrier because it increases the risk for red-green color blindness. Now, red-green color blindness is not a life-threatening condition. It increases risk because of that failure to differentiate colors for different scenarios, but otherwise is probably not a reason not to conceive or have a baby between those two partners. In other types of conditions, that information might be more valuable, and we'll talk about what I mean by that in just a second. So if mom offers a normal color expression and the Y chromosome is offered by the male, then what we've got is a male, essentially a male that has normal. I should reverse that. We have a male with normal vision. Okay, so the way we're going to get a male that has color blindness that might be a surprise is that the mom who was a carrier offers the recessive expression of color blindness, and because the Y chromosome doesn't carry a match, we're going to have a male who has abnormal vision. In other words, they're red-green colorblind. So the scenario where a female expresses red-green color blindness is going to be a known possibility, because in order for that to occur, the male allele has to be recessive, right? Because it's the only one he's got to offer. If this individual male here decides to reproduce at a later date, and he reproduces with a female carrier, and the female carrier contributes the X small c allele, then that individual can become colorblind, right? The Y is not carrying an allele that matches, right? So that scenario would be a surprise, because you assume the female had normal vision as far as everyone was concerned, and it's not a type of situation where there can be some sort of partial expression typically. So this would have been a surprise to those parents as far as that goes. Now, what's often used to try to explain some of this inheritance is what's called the Punnett square. So some guy named Punnett came up with this mechanism. And what that does is take a look at what the parent's genotype is to be able to predict what the genotypes of the offspring would be. So in a scenario where we form a square and we have what's sometimes described as typical or simple Mendelian inheritance, so that guy, that monk, Mendel, that experimented with pea plants and really sort of introduced this whole idea of genetics and the contribution from parents, is if we put the male on the vertical axis and the female across the top on the horizontal axis, then she's going to be double X, he's going to be XY. Okay, and if we're talking about the expression of some simple dominant recessive sort of arrangement, if we had, let's go back to the letters A. So if we have capital A's across the top, that mom is dominant homozygous. And if for this particular trait that we're following, the dad is heterozygous. Why did I do the letter H? I want to do the letter I there, don't I? That's just going to mix us all up. I mean, the letter A. So now when we put combinations together, we can see that up in this upper left corner, we're going to have two X's, and across two X's. And when we come here, we're going to have an X and a Y and an X and a Y. So when we talk about the autosomal traits of a male and a female presentation, here we're going to have both capital A's and both capital A's. So they're going to express the dominant gene. Let's keep it simple and say that that's brown eyes, right? Let's ignore the fact that we have multiple contributions to eye color, but just to keep things nice and simple. So down here in the male offspring, we're going to have dominant and recessive, and then we're going to have dominant and recessive in these possibilities. But in all cases now, all offspring are expressing a dominant gene, so all of these offspring will be brown-eyed individuals. If we changed it and looked at a mom who carried the blue trait, but she's got brown eyes based on dominance and recessive, and dad was also brown-eyed, so I'm designating the dad as an XY, but we're talking about autosomal genes, just so that we're clear here. We're not talking about the sex genes just yet. We're just identifying the male contribution and the female. So across the top, we're going to have a female who becomes homozygous dominant. We have a female who is heterozygous dominant. And now we have a male who's homozygous recessive. Oh, it's not homozygous recessive in this one. Heterozygous, so that it's a male carrier. And this male will have both recessive. So that makes this guy ends up with blue eyes. He expresses blue eyes. But everywhere where there was dominant, brown and brown. And this one would be clearly brown. So I hope that's starting to make some sense. Now, where this becomes significant is when we have what's called an X-linked recessive gene. So in an X-linked recessive gene, if it's described as being X-linked, it's on the X chromosome of the pair of the sex chromosomes. So it wouldn't be present on the Y chromosome. There would be no match on the Y chromosome. So in a male who is X-linked recessive, so we pulled a small letter H in here, and there's no match on the Y, this is going to be expressed. This condition, whatever the small h represents, is going to be expressed. And one of the conditions that becomes significant, one of the things that we worry about in healthcare, is a condition called hemophilia. Hemophilia is an example of an X-linked recessive gene. It can be life-threatening. It often causes severe debility and shortness of life overall if it's not recognized and not treated effectively. And even when treated effectively, sometimes it can be so detrimental that it has a very detrimental or very significant implications with respect to quality of life and sometimes even quantity of life. So this would be called an X-linked recessive disease. If the disease is significant enough, it may influence your choices for reproduction. So in this case, the recessive gene is going to be expressed because there's no match on the Y chromosome. So if this is an abnormal gene that presents on the X chromosome because there's no match on the Y, then in males, it's going to present itself. So in this case now, what we would be looking at is, let's look at hemophilia. So this is X-linked recessive inheritance. So our X chromosome offered by the female across the top, and our X and Y chromosome across the bottom. So if we had a male who was normal, we're going to do a big capital H. And if we had a female who was a carrier, she may be unaware that she's a carrier. So she would have one dominant and one recessive allele for the trait of hemophilia. She will go through her life with no complications because hemophilia is a recessive condition and she has a dominant allele that prevents the expression of this particular disease, hemophilia. Dad is also normal because he has the dominant form of the non-expression for hemophilia. So the females are going to be born, possibilities are going to look like this. One homozygous, normal, one heterozygous, normal female. When we look at the male possibilities, there is no match on the Y chromosome, so the dominant expresses with no interference. But on our second possibility, we've got our recessive gene now with no match on the Y chromosome. So the recessive gene is going to express. This is our patient now. That is going to express hemophilia. This is a blood disorder that prevents proper clotting from occurring. So they're exposed to bleeding episodes. And they tend, the blood cells are very fragile. They fracture easy. So sometimes even to something as simple as running can cause breakage or fractures of blood cells in the joints. they bleed into the joints, they cause joint pain, and worse than that, they interfere with oxygen delivery and carbon dioxide removal, so it can become life-threatening very quickly. So this explains how X-linked recessive inheritance occurs. Now, what I want to do is show you are these same things, but in a more physical, a more visual way. Let's go down here. Let's blank that one out and go down here and look here. So in this, let's find a color that's going to show up. So in autosomal dominant, simple Mendelian inheritance, both parents are going to present like this. So when the autosomal pair, those 22 pairs, have an allele that is dominant in one or both parents, then these are the possibilities. So in this example, we've got a parent that is a carrier of some sort of mutation, some sort of recessive expression. So in this example here, the light blue allele represents normal, and the mutation is a purple. So dad is affected in this situation with a dominant mutation. Okay, that's what this is trying to show, autosomal dominant mutation. That's why dad is all purple. He's expressing this presentation here. Now, lots of times these dominant expressions are part of our normal traits. But if this is describing a pathology, what we would look at is see dad is going to contribute his chromosome to the male and to a female. Then the male is going to be affected and the female because it's dominant. But he's also heterozygous, so he's got the unaffected gene to contribute as well. And he can contribute to the female or the male with his recessive gene, and you would have a normal expression there because mom is not expressing the mutation. Dad is expressing the mutation. If the mutation is dominant, then only one allele needs to be present to be expressed. So this is a situation where the mutation was dominant. And so anywhere where the mutation is presented is going to be expressed. Now, this is a smaller picture here, but when the autosomal trait is recessive, dad is a carrier, mom is a carrier in this example, carrier, carrier. So there's a possibility that one pair from each that is recessive, or dark blue color, is going to be expressed now because mom and dad both had the capacity to present the affected gene. If they also both present unaffected, then we have the scenario where the child is not going to be affected and not be a carrier. In these two examples here, where there's a contribution from the recessive gene and dominant on this side, a carrier but no expression. And in the male situation, the same idea, a carrier but no expression. The recessive way of looking at this is this one. So in this case, we see that the dad is unaffected. He has the dominant allele, so he's not going to express. So this is a guy, he would have the let's see, male has the dominant allele. So he is unaffected. So if we're talking about X-linked, our best example is the one that we described on that other slide, hemophilia. So the male has a dominant allele. Mom is a carrier. So when mom's a carrier, we describe this as being an X-linked recessive allele. She's a carrier, she's mixed. So her dominant allele makes sure that she does not present with the pathology. but she is a carrier. So it's possible then for her to pass on this recessive gene to one or more of her offspring. So in our Punnett square, we could see that a percentage of the male offspring is likely to become affected. So, and that's our example right here. Because the X-linked is recessed and there's no Y chromosome to match, then as long as the recessive gene from the mom is present, then it's going to be expressed. So the male son is going to present with hemophilia. One of the daughters in this example is not affected, but is a carrier. And in these situations where the male X chromosome presents with the female's X chromosome that is unaffected, you're either going to be homozygous male or homozygous female in that scenario. And so the idea here is when you take the Punnett square, which is all we're looking at, is it's the same idea. Okay, we had the female across the top and the male across the bottom. There's nothing to match up. So a normal male would present this way, carrier female would present this way. When we combine the chromosomes, we get a homozygous female. That's this female right here. We get a heterozygous female who is a carrier. So that's this daughter right here. And when we combine the XY offspring, we're going to either get a normal presentation for the blood, in this case, the non-hemophiliac, but if the recessive presents, we get the presentation of the disease, the affected son. So this is our affected son now in this situation. Let's try and do a bit of a color code here. So our affected son is this individual right here. So that would be described as an X-linked recessive condition. So this becomes significant from a genetic counseling point of view. If you determine as the female that you are a carrier for hemophilia, you may choose to have some sort of intervention if you're interested in having children. But you also may choose not to have children because you're concerned that there's a risk that any male progeny, any male children that you were to have could end up with hemophilia. So it may affect your options or your choices for family planning in that respect. Let's see, where are we here? We want to get to a layer we haven't drawn in yet. All right, so what do we do to create genetic variability? Well, we have talked about this already once, one component of it, but what we want to be aware of, and this is our final topic in this particular subject, genetic variability can be is basically what we're saying here is that's going to give us different genotypes, which is what's going to give us different expressions of those genes. And that's why we're all going to look a little bit different from one another. And it's based on a couple of things. So we create this genetic variability a number of different ways. And one of those ways is through what's called homologous recombination. So that sounds kind of fancy. And that's what occurs in meiosis, particularly in meiosis 1. The first phase of meiosis is where the homologous chromosomes, the same paired chromosomes, are going to cross over and exchange some of their material in basically a random or variable way. So this is a situation where in meiosis, particularly meiosis 1, the homologous chromosomes they cross over and exchange genetic material randomly. So every time a gamete is introduced to meiosis, spermatogenesis, oogenesis, there's going to be this genetic variability introduced where the chromosomes cross over. So that means that some part of the chromosome from the male and the female parents of that individual are crossing over. And then some of that material is exchanged. It may change position within that chromosome. So that's going to create some variability. The other way that we can create some variability is, especially in this day and age, when we have so many readily available means of changing where we live, we either move into a new area or out of an area, and that creates genetic variability because now what we're creating is movement of genetic pools, essentially. So you're going to move from one part of the world to another part of the world, and that's going to create some genetic variability. So you can appreciate that if you were, say, have a long, consistent heritage from the Greek population, that there's going to be a little bit more similarity amongst that population because of the chances and probabilities of similar genetic material being shared in that population. So if you move out of that population into a different population, you move from Greece to Canada, then there's a greater chance that there's going to be some genetic variability established between partners from different population groups. So that's another way that we get some genetic variability. And the final way, which again sort of supports what Darwin was all about, is we talk about genetic mutations. Now, in genetic mutations, they can be either positive, they can be negative, or they may be neutral in terms of their benefits. But this is where some of the terminology comes from Darwin and Darwinian theories and so on. He talks about natural selection, and he talks about survival of the fittest and those sorts of things. So when a genetic mutation is considered to be positive, it increases your chances or your condition for survival. So all of us who live today are products of multiple types of genetic mutations that have occurred over the last, several million years of our awareness of being and what we know. So this DNA mutation or damage occurs on a daily basis throughout our lifetimes. And we have multiple ways to regulate that. Typically, if it's a genetic mutation that's positive, it may be left alone. It may be a strong enough presentation that it isn't recognized as being negative. When a genetic mutation is negative, we have chemical messengers in our body that can recognize those negative mutations and cause that cell either to die, and when it happens internally this way, it's got a fancy word that's called apoptosis. I'll write that down. But talking about genetic mutations, let's carry on our topic of genetic mutations here. Remember, we said they could be positive, they could be negative, or they could be neutral. And the idea is that historically, positive mutations that increase our chances for survival and is generally described as a natural selection process, is what we are the products of at the end of the day. So when a mutation is negative, or even in some cases neutral, then it can be repaired. So there are chemical messengers within the nucleus of our cells, or sometimes the cytoplasm of our cells, that will be activated as a result of a mutation. And if that mutation is harmful, one of the results of these attempts to repair is going to be apoptosis. So, for example, there's a gene that expresses a protein that's called expresses a messenger, a protein called p53. Most of us have normal presentations of this protein called p53. This is a gene that ultimately regulates the cell cycle. So it regulates cell cycle. In other words, if a cell undergoes mitosis on a regular basis, this p53 molecule is going to sort of watch and make sure that this expression is normal. And so in normal cells, p53 is relatively low in numbers, but in abnormal cells, it rapidly increases. And the result of an increased level of this p53, which is just one example of the way that we can regulate mutations that occur, is going to give us a couple of possibilities. So the first thing that the higher expression of p53 does is it's going to seize mitosis. So it's going to stop further reproduction of this abnormal cell. And then the second thing that it does is it's going to initiate repair. So if the chromosome mutated and that mutation is potentially harmful, which happens, for example, in certain types of cancers, then this expression of p53 will initiate chromosome repair, try to fix that mutation. If none of that can happen, then the third responsibility for p53 is that it's going to initiate apoptosis, which is a type of programmed cell death. So this is part of normal physiology. And as I mentioned earlier, these genetic mutations happen to the tune of thousands per day. Thousands, tens of thousands of mutations occur in the day. And if it's a negative or sometimes even a neutral mutation, then the nucleus of the cell that's regularly low in p53 the gene starts to express more p53. And then that's going to result in one of these three or all three of these responsibilities for p53. Stops mitosis, that's going to give us some time. Initiate the chromosome repair. If that chromosome can't be repaired, then what the third thing may occur is cell death, programmed cell death, otherwise known as apoptosis. It's a type of cell suicide. It's neat and tidy. It doesn't generate an inflammatory response or anything like that, so you're not even aware that it's happening. So p53 is sometimes described as the guardian of the genome, guardian of our cells, guardian of our genetic expression. It's also known as a tumor suppressor. You'll talk more about that when you talk about cancer, when you talk about pathologies. But tumor suppressors are going to do just what it sounds like they do. They slow down the growth of some sort of abnormality, which is what cancer is, rapid growth of cells that no longer sort of respond to normal checks and balances in the body. So some cancers we know of occur in people who don't have properly expressed p53. So treatment mechanisms can be implemented with this understanding. And that's part of the research on cancer and explains why some people are more at risk for some certain types of cancers. Okay, so that sort of covers the basics for inheritance. You want to be aware of terminology like dominance and codominance and recessive, heterozygous and homozygous. Be aware of the Mendelian square or the Punnett square and how it can be used to predict the presentation or the absence of a disease in offspring. And you'll have the basic understanding for genetics and inheritance for this course.